So that's objective one. Let's move on to objective two. We're going to describe the communication across the chemical synapse, and we're going to explain how neurotransmitters are released and talk about their action after they're released. So let's talk about the anatomy. We're going to draw this out just to make it a little bit simpler, but let's talk about the anatomy, the structures that are involved first. So we've got these two neurons that are in series. We've got the presynaptic and then the postsynaptic. We've got the neurotransmitters that are within the axon terminal. So if we zoom in closer to the axon terminal on this presynaptic neuron, we'll see that there are these vesicles. They're called synaptic vesicles, and they're storing neurotransmitter. Now, the type of neurotransmitter will differ depending on the exact type of junction. That's why it's important to understand that this junction can be different. If it's a muscle cell, cardiac muscle versus skeletal muscle, the exact neurotransmitter will differ. But for just the sake of explaining the event, let's just think of this as X neurotransmitter. So we've got these synaptic vesicles in the axon terminal with neurotransmitter X. We've got these voltage-gated channels lining the axon terminal. So if you remember back when we talked about the different types of channels found throughout the neuron, we said at the axon terminal, there were voltage-gated channels and not voltage-gated calcium channels and not voltage-gated sodium or potassium channels, the way that we see along the axon. We've also got this reuptake molecule that's going to be important as far as recycling any excess neurotransmitter that's left in the junction. And I'll talk about why that's really important. We've also got the synaptic cleft itself. So the space between the adjacent neurons is called the synaptic cleft. We've got the receptor on the postsynaptic side. So in order for the signal to be transferred from one neuron to the other, there must be a receptor onto which the specific neurotransmitter in the vesicle here can bind. So there must be this specificity between this receptor and the exact neurotransmitter that's being released. We've also got enzymes that are studying in the membrane. We're going to talk about the exact name of this enzyme later on, but the purpose of this enzyme is to help degrade or break down any extra or excess neurotransmitter that's lingering here in the synapse, in the junction. And so these two molecules, the enzyme here on the postsynaptic side and the reuptake molecule on the presynaptic side, the goal of these two structures is to clear the junction after this synaptic communication takes place. And that's really important because we need to cease this signal when the signal has ended. There needs to be clearing of the junction. Otherwise, this will continue to communicate indefinitely. And that is not what we want, right? We want to communicate this neurotransmitter, and then we want to clear the junction so that this signal can cease. So that's kind of the overall objective here at the synapse. Those are the players that are involved. And so let's actually walk ourselves through the events or the process that's involved as far as synaptic communication. Okay, so here we're looking at synaptic communication. And so we're really going to zoom in on our synapse. So essentially we've got this presynaptic neuron. Here's our axon terminal. And then let's zoom in further on the adjacent cell. So we know we've got our myelination. Let's say this is a myelinated neuron. We've got these voltage-gated sodium channels that are condensed at these nodes of Ranvier. And so we've got the action potential leaping down the length of the axon. And when it gets here to the axon terminal, it's actually going to trigger the opening up of voltage-gated calcium channels. So we've got this voltage-gated calcium channel here on the membrane. Let's draw this in. And so again, it's just like the voltage-gated sodium, it's going to be sensitive to voltage. And so when the action potential gets to the axon terminal, this is essentially going to depolarize the axon terminal, and that will open up, because of that voltage change, these voltage-gated calcium channels. And as you would expect, calcium is going to rush into the cell, rush into the terminal bouton here, the axon terminal. Terminal bouton is another term that you may hear referred as far as the axon terminal here. And so when calcium rushes in, calcium's got two charges. When calcium rushes into the axon terminal, we're going to see the release of these synaptic vesicles. So let's bear in mind, we've got these vesicles in the axon terminal. And they've got this specific neurotransmitter inside of them. We'll talk about what some of these examples are later on. And so when the membrane potential changes down here, calcium rushes into the cell, and the rush of calcium into the cell is what triggers the process called exocytosis. If you remember, we talked about transcytosis as a part of our membrane transport mechanisms. This is an example of exocytosis. So this is where the membrane of the vesicle fuses with the membrane of the axon terminal and then dumps all of this neurotransmitter out into the cleft. And all of that is triggered by the rush of calcium into the axon terminal. What happens at this point is we have binding of that neurotransmitter to our receptor. So on the adjacent cell, we've got a specific receptor here that's going to specifically bind this neurotransmitter. Right, so let's say we bind this neurotransmitter here. That is going to generate some type of cellular response, okay? This is going to bring about some type of cellular response on this cell. And I'm going to talk a lot more about that process later on. So kind of stay tuned here. How this cellular response is brought about is going to differ depending on the type of receptor, the type of neurotransmitter, et cetera. But the binding of the neurotransmitter to this receptor is what brings about our cellular response. And this type of response can be one of two things. It can be a fast response, or it can be a slow response. And I'll talk about what that means a little bit later on. So this can be a fast response or a slow response. After that response is generated on the adjacent neuron, this is where our two molecules come into play. So the first molecule we said was an enzyme on the postsynaptic membrane. I like to think of this enzyme as being a little Pac-Man. So I'm going to draw it like a Pac-Man. Let's redraw that. This is called acetylcholinesterase. And the purpose of acetylcholinesterase, so it ends in ASE, it's going to degrade or chop or break down any excess neurotransmitter that's lingering in the junction, and those degraded pieces can be reuptaken by the reuptake molecule. So if you remember, we talked about this reuptake molecule on the presynaptic side. So this is our reuptake molecule. Okay. And so after the acetylcholinesterase degrades the excess neurotransmitter, then that can be reuptaken back into the presynaptic neuron and essentially recycled. So I'm going to put in brackets here, recycle. Okay, so the purpose of the enzyme is to break down excess neurotransmitter into smaller pieces. The purpose of the reuptake molecule is to reuptake those pieces into the presynaptic neuron, and they can be recycled to make brand new synaptic vesicles. Now, there are actually three methods for clearing the junction of excess neurotransmitter. The first one is that we can have the excess neurotransmitter diffuse away. So that's an option as well. We can have excess neurotransmitter diffuse away. The second option is that we can have the acetylcholinesterase break down the excess neurotransmitter, and then those smaller pieces can be reuptaken. And then the third mechanism is that we can just have these large neurotransmitter pieces reuptaken as they are, okay? So over here, I'm gonna put three options for clearing the junction. Okay, so they can simply diffuse away. They can be degraded and then uptaken or reuptaken. Oops. Or they can be reuptaken as is. Okay, so that's the purpose of this reuptake molecule, is to basically swallow the excess pieces or the neurotransmitter as is, and uptake these molecules back to recycle them to make more. Okay, any questions here? Any questions at all? I also want to make a note that this is exocytosis. So the process of dumping the neurotransmitter into the cleft and the fusion of the synaptic vesicle membrane with the membrane of the axon terminal, that process is called exocytosis. And again, it's triggered by the rush of calcium into the axon terminal. Okay, any questions here before we keep going? Any questions? And we're going to clarify this again and again. We're going to talk about this several times. So make sure that you understand the sequence of events that are taking place here. We're going to talk a lot more about this fast or slow response in a bit as well. We're also going to stress on why it's so important to clear the junction. Okay, that's a really important concept. You must clear the junction, otherwise we can get a lot of different pathologies. We'll talk about some of the clinical examples like myasthenia gravis and different types of paralysis or muscle disorders that could exist if we don't clear this junction effectively. So the importance of the acetylcholinesterase and the reuptake molecule is so that after the signal has been sent, the junction is cleared completely. So someone is asking, is the voltage gated sodium channel from the action potential? And that is, yes, that's correct. So this voltage gated sodium channel is the exact one that we talked about that initiates the action potential, that is responsible for the conduction of the action potential down the axon. Okay, and then that signal is going to be converted to chemical energy when it gets to the axon terminal. This chemical or neurotransmitter is what's going to pick up the signal, carry it across the junction, bind to this receptor, and then we're going to talk about the events thereafter. Okay, so let's keep going here. So this is a summary of those exact events. We've got the action potential traveling down here again, just to kind of repeat this so that we're getting more comfortable with this. We've got the action potential traveling down. It's kind of step one. It triggers the opening up of this voltage gated calcium channel such that calcium can rush in. Calcium rushes in and triggers the vesicle docking and secretion. That is essentially exocytosis. So the docking of this vesicle with the membrane of the axon terminal itself and the emptying of the neurotransmitter into the junction. It will travel across the synapse, and I'm using the word synapse and junction interchangeably. So just to avoid any confusion there, those are the same meaning. So it travels across the synapse here, binds onto this receptor. Again, it's going to be specific to the neurotransmitter. That will bring about some type of cellular response. And then the response will be terminated when all of that neurotransmitter has left the synaptic cleft or has left the junction, okay? And so to bring that about, we have the enzyme that's on the postsynaptic side, degrading any of that excess neurotransmitter into smaller fragments. And then those smaller fragments can either diffuse away or they can be reuptaken, or the neurotransmitter as they are can diffuse away or be reuptaken as well. So there's a bit of variation as far as exactly how they're cleared. There's a few options there. As we can see here, they can be reuptaken in their full state, they can be degraded and then reuptaken, or they can be cleared away, diffused away from the junction, okay? But the essence here is that there's an option for them to be reuptaken back into the presynaptic side and recycle to make some more neurotransmitter, some more synaptic vesicles. Now, there's a slight delay here. So this is not an immediate response. There's a 0.5 to 5 millisecond delay between when the action potential gets to the postsynaptic membrane and the membrane potential changes, and when we actually see some change on the other side, okay? And so it's caused by the changes that are associated with the exocytosis process. So the docking of that vesicle onto the membrane, the release of neurotransmitter, and the time it takes for that vesicle to dock. What it's not related to, right? This is kind of what we would maybe kind of think, but it's not related to this process, is the time it takes for diffusion to happen across the cleft. So let me kind of say that in another way here. So the slight delay that we see between the action potential getting here and there being a membrane potential change on the other side is due to the time it takes for the synaptic vesicle to dock on the membrane of the axon terminal and for that neurotransmitter to be released into the junction. The delay is not due to the time that it takes for the neurotransmitter to diffuse across, okay? So just to kind of clarify the cause of that synaptic delay.